Hello, College Biology. This video is about mutation, and in it we will discuss the changes in the nitrogenous base sequence of DNA, because that is what a mutation is. So, how do these get started? First of all, the radiation is a big dang starter of mutations. This is energy traveling in waves, and it can hit those particles, and it can damage the nitrogenous bases, or hurt nucleotides, or rearrange particles in the nucleotides so that the nitrogenous bases change. We're talking about examples like UV radiation and X-rays. We also have mutagens, or chemicals that will affect your DNA, like smoke and tar and aspartame, and apparently some common ingredients in acne products that I don't know anything about, and nitrate and nitrate preservatives for meat processing, etc. And viruses. We know that viruses can get in there and inject their DNA into yours, so they can change the nitrogenous bases of your DNA. And there's another one that isn't from an external source but happens internally. We copy our DNA during DNA replication for cell division, and errors can occur during that process, and if they miscopy the DNA, well, then that error will stick with all of the cells in that generation, and all of their subsequent progeny will always have that mutation. So mutations can get started a couple different ways. Now, once we accept that there are mutations, we have to consider what are the types of mutations. Well, one type of mutation is a point mutation, where one nitrogenous base is affected. You could have a sequence here, and then one of those nitrogenous bases changes to another. We can also have frame shift mutations, and frame shift mutations will change all of the codons after the mutation. And there's two subcategories of that. We can have an insertion, where we add a new nitrogenous base, and so that shifts everything down one, or we can remove a nitrogenous base, in this case thymine, and that will shift everything up one. So what does that mean? Well, what does that mean for the consequence of mutations? Well, there are some mutations. I think I just lost one of my slides. Well, there are some mutations called missense mutations, where one codon is changed, so one amino acid changed, which means this protein is going to fold differently. And most likely that folding is going to result in this protein either being unable to do its job or to do its job poorly. It's the most common thing there. Now, there's also nonsense mutations, where often they can be caused by frame shift mutations, where they shift a ton of the amino acids, and this can cause complicated problems. Both of these point or missense and nonsense can, if they affect a stop codon or a start codon, you could have a protein that just doesn't get formed at all, or you could have a very, very different protein. Now, one of these slides, I think I deleted it. Yeah, I totally deleted it. Was a slide that shows that sometimes you can have a mutation that occurs, except that the codon that it changes to will bring the same amino acid. In that case, you have a silent mutation. You have something that isn't going to change at all because the protein is using the same amino acids. Furthermore, if a mutation occurs in a non-coding region of DNA, that's a silent mutation because the DNA has changed, but no protein is affected, so it's irrelevant. So what does this mean for organisms? Well, the most common consequence of mutations, because silent mutations are real, mutations occur that do not affect protein, because there's, a lot of non-coding region. The most common thing is that the organism is not affected. Right there. Look at him. Boring day. Everything's fine. Just chilling out. Mutated. Doesn't matter. I don't even know about it. Nobody knows. Now, there are other consequences. If the mutation is really important, it could cause death to the organism. And if the mutation is in a protein that has some external visible quality, you could see a change in traits of that organism, or a change in the biochemistry of the organism, which might be tougher to see, but could be detected by chemical means. And in extravagant cases, when an entirely different protein is made, you could have something amazing. Like in this fossil of the Tiktaalik, it is a fish, the oldest organism known, that has the ability, with its fins, to do a push-up. That's right. this organism is the oldest organism that has the right structures to do a push-up. And this is the forestructure of the legs as we know it. Organisms older than this, they don't have legs, they don't have arms. But organisms after this, they do have legs, they do have arms. So we see an amazing, amazing new trait as a consequence of a mutation here. And then you can also have loss of traits due to mutations. This is an ancient snake skeleton, and you can't really tell, but well, you can actually. If you look over here, you can see little leg structures, but this is pretty far away. So let's zoom in. Boom. Leg structures right here as part of the snake skeleton. This fossilized snake had legs, but modern day snakes, not so much. So there was a mutation at some point that contributed to the loss of this feature in this organism. Now, so many of the structures that we have are not actually contributed by one mutation or one gene. It's actually usually a lot of genes cooperating together. So sometimes mutations subtly change an organism, but when mutations build up regarding a particular trait over time, you can have a drastic change, addition, or loss of that trait. This video has gone long, so we won't do a review for this video. We will do that later, and so the next video will be a review, and I hope this helps struggle well.